time. So we'll check in about projects, because I know Shay, you may have replied, but I didn't get to it. So okay, you weren't feeling up to it either. Okay, so anyone who's actually here today that doesn't definitely have an approve for your projects, check with me before you leave. Okay, so I just posted the section 5, and it is right here. So for this course, we really need to focus in on eukaryotes, and usually that's what we care about because that's us, right? But bacteria, which are prokaryotic, have a different way because they're a little bit different from us, right? You could definitely say that. So we will look a little bit at it, but I'm not going to test you guys on that. I'll test you on section 5, which is as it relates to the chromosomes of eukaryotes. So in this chapter, we open up with, I didn't get rid of the animations. That's going to annoy me. I'll just put it all up there. So chromosomes contain the genetic material, but they're not just DNA, right? They are DNA and protein, and sometimes a little bit of RNA, depending at what phase of the cell cycle you're in for those cells. So for bacteria, as I said, they're circular, right? So they're one strand of DNA usually, and it's circular. It's a closed loop. Unlike most eukaryotes, and again, this is most for both, right? We're saying typical, most. Are there exceptions to this? Absolutely. There's a bacteria that causes Lyme disease that has linear chromosomes, right? That's abnormal for a bacteria. Although I have to say I don't know any eukaryotes that have circular chromosomes. For the most part, eukaryotes are going to be linear chromosomes, and they're going to be found within a membrane-encased organelle we know as the nucleus. That's one of the defining characteristics of eukaryotes. Where prokaryotes, remember, don't have a nucleus, and that's what bacteria fall under. But all cells have DNA, right? We've already proven, right, this is the genetic information. We know how it's organized at the molecular level, and we're going to go a little bit larger. Now, of course, for eukaryotes, their mitochondria, as we've already discussed, also contains DNA. For plants, their chloroplasts also contain DNA. But we refer to those genomes separately, right, as mitochondrial and chloroplast genomes. Now my nose is going to start acting up. So it stores the information, right? We know the base sequence, we know the base pairing, we know how the nucleotides bind with each other. How do they bind? What's the name of that bond between nucleotides? Between that sugar and the phosphate? Phosphodiester bond. Phosphodiester bond. Right? Yeah, I would learn it before the test. Hint. Phosphodiester? Oh, when we get through the stuff. So I'll have to look. It's not next week, it'll be the following week, whenever we get through chapter 11. We won't get through it next week, so, but definitely the following week, whenever that is. Okay, so for DNA, right, it needs to carry the sequence also for the RNA, which we're going to talk about this in chapter 11. I think. Again, I'm off today. But we go from RNA to proteins, right? To the cellular proteins that are actually the workhorses and do most of the work in the cell. So even in the DNA is how to make the messenger RNA to make the proteins that are going to combine with the DNA to form the chromosome. So, of course, replication has to happen. These have to separate, and we'll talk about that a little bit, I think. Or are we going to skip over that? We might skip over that. You guys have done mitosis and meiosis, right? We don't have to do that in this class, right? And compaction, and that's what we're going to focus in on this lecture, of those chromosomes. And they give you some numbers in here. I don't want to misspeak, so I'll wait until we get to that, and it really helps you with it. So this is for bacteria. This is one of the pictures that I kept in from the bacterial section. So remember, circular chromosome, right? So this is DNA, right? The alpha helix that's just showing it as a single ribbon. The first level of compaction involves DNA binding proteins, and they form these loop domains. So we can make this loop smaller, right, by having it kind of loop up. And how it does that is it binds to these DNA binding proteins. And then the next level of compaction that bacteria do, and so for this level, from this circle to the loops, is 1,000-fold compaction in the size, right? So imagine that, you know, this is 1,000 meters. We just shrunk it down to 1 meter, right, by forming those loops. So I guess I'm trying to think that, like, comparison to our everyday lives, right? You know how, like, you take a garden hose and you wrap it around, right, and you loop it up, right? It takes up less linear space when you loop it. Does that make sense, right? So when you have it stretched out in the yard, think about how long it is. Maybe you've got a 50-foot hose, right? But if we loop it around itself, right, then we can compact it into that smaller space. We no longer need 50 feet. We can maybe compact it down to a single foot, right? You guys with me? Trying to put it. So there's our loop domains. So as a 10-fold, that's actually 10-fold. From circular to loops, it's a 10-fold. But we get it compacted a total of 1,000 times to actually kind of fit in the cell and not be stretched out. So obviously, if I don't have this memorized, remember I said this is the stuff I'm testing you on. So for an example, E. coli has about 50 to 100 of these loops. And within each loop, there's about 40,000 to 80,000 base pairs. So we're talking about A's, G's, T's, C's, right, within each one of those loops. And of course, this is going to vary from species to species depending on how much they have. Now, the next level that they do is then they kind of twist these loops. And so, again, the best real-world example I can give, if you take a rubber band, right, that's circular, it's like a loop domain, and then you twist it on itself again, right? And so that's what this is representing here. But they do it a little bit more at the molecular level. They'll even literally nick the DNA and twist it upon itself, right? Because remember, it's a helix itself. So in order to be able to twist it, you've got to kind of distort it a little bit. And there are special enzymes and processes involved in what this is referred to as. And this is called supercoiling. And so, I want to see, and I'll post it if it is helpful. Let's see. This is an animation they have of the chromosome compaction. Oh, that's the gyrases for supercoiling. I'm not going to worry so much about that. Let's see if this is at all helpful for us as a visual aid. So let's see.
About 1,000-fold to fit within a bacterial cell. Part of this compaction comes through the formation of loop domains, where segments of DNA are folded into loop-like structures that are held in place by DNA-binding proteins. The formation of loop domains compacts the DNA about 10-fold. Further compaction is accomplished through a process known as supercoiling. In this process, which is under the control of the enzymes DNA gyrase and topoisomerase 1, twists are introduced to the DNA molecule that cause it to compact by coiling onto itself. This is much like the process of coiling that you can observed by twisting a rubber band. DNA looping and supercoiling make the bacterial chromosome more compact so that it can fit within the nucleon of the bacterial cell.
I don't know how helpful that was.
Also kind of reminds me of those telephone cords that get wrapped around.
Oh, old-time telephone cords, definitely. Right, yeah. You can definitely imagine some major supercoiling there when you overstretch those babies. Yeah, I don't even know. Like, my son has no idea what that is, what a telephone cord is. No idea. I remember that, stretching it across the house. But yeah, that's a common analogy that scientists have been giving for a long time, is the rubber band. So I should bring me some rubber bands in so we can twist them up, though. Okay, I don't know that's in your book, right? If you know how to get to the animations in your book, I don't know that I'll bother posting that for you guys. So this is the section that we need to focus on, and so we need to be able to define chromatin. We need to outline the structure of the nucleosomes, the 30 nanometer fiber, and the radial loop domains. And remember, this is to eukaryote. So this is our next section. This is structure of eukaryotic chromosomes in non-dividing cells. So they estimate that if you stretched, now think about this is molecular, right? So we're talking about teeny tiny little things. That if you stretched end to end a single set of human chromosomes, right? So that's only 23, right? Chromosomes, single set. We're not talking about even diploid. Here we're talking haploid. From end to end, if you stretch them out, it'd be about 1 meter long, which is about a little over 3 feet, right? Or about a yard, a little over a yard. Yeah, look at that. That, something, take a string that long, right, is inside each one of your microscopic cells. It's kind of crazy, right? So again, I mean, like, think of that 50-foot hose in your yard, right? You've got to compact that inside the nucleus of a cell. So the cell's nucleus itself, right, when you measure it under the microscope, is about 2 to 4 micrometers, which is, did we look at the scale thing in this class? We did not look at that, did we, in this class? Okay, so this is a really cool website. Let me see if I can find it. It is, I think it's Utah. Genetics, cell size. Watch it not be Utah, watch it be another state. It's actually their genetics website. Yes, Learn Genetics. A student showed this to me. This is really cool. So if you're in my micro class, you've already seen this because I've been using it for a couple of years now since I found it. But so here it's grayed out. That's not cool. Why do they have it grayed out? So here's the scale. So a meter, right, and centimeters are a meter divided up into 100 units, right? Most of us have dealt with centimeter rulers, right? And then a millimeter is 1/10 of a centimeter, which is 1/1000th of a meter. So again, we can still see that, right? You see those little lines on a centimeter ruler. I'm trying to think. They don't have one on here for this. Microbiologists measure in micrometers, which are 1/1,000,000th of a meter. Or as if you compare it to millimeters, it's 1/1000th of a millimeter, right? So imagine those little lines on a centimeter ruler, right? And cutting that into 1,000 slices, right? That's how small we can see with our microscopes. In truth, we can see about 200 nanometers, right? Which is even smaller than micrometers. So nanometers is 1/1000,000th of a meter, or 1,000th of a micrometer. So we're talking really small measurements here, right? So this thing is really cool because it's interactive. So notice a coffee bean, right? This looks even kind of big up on our screen. But they give us the size. So it's about 12 millimeters by 8 millimeters. So you could measure that with your little centimeter ruler, right? Grain of rice is starting to get kind of small though, right? Unless you've got really good eyesight, or sesame seed. But as we zoom in with this program, right, you could see, right, that you're talking, you know, really small, right? Like I said, you could barely probably maybe measure a grain of rice with a ruler. You're really not going to actually get measurements with these guys until you use a microscope. So a grain of salt you can actually see under the microscope, right? And that's actually quite large as far as microorganisms go. That's a half a millimeter. So again, we're still within that range of your ruler, per se. When you get to something like an amoeba, which again is really big in comparison to a bacteria cell, which is much smaller. So these guys are now we're getting into micrometers, right? So this guy's about 500 micrometers. Paramecium, right? We're talking about 200 micrometers in length. About the human egg, which is the largest cell in a woman's body, right? Because they make all the organelles and everything for the growing embryo. That's about 130 micrometers. And then look at this poor little spermy guy down here, right? Just this mobile DNA. Look at him, right? He's longer than he is wide. And so then look at here, like, so here we have, this is really large structure in our eyes, the photoreceptors. But here's a skin cell, right, a red blood cell, and look at chromosome compacted, the X chromosome even, compacted, 7 micrometers, right? That's bigger than this poor little E. coli right here. One of our chromosomes is bigger than one of these guys, right? It has a mitochondria, right? And this goes all the way down, right? Viruses, ribosomes, all the way down to the core atom, the carbon atom, right? All organic matter is based on carbon, right? All have carbon at the core. Right? And then you have hydrogen and oxygen, and some of them we know we have nitrogen and sulfur, and what molecules that make up our body contain sulfur? So what is methionine? It's an amino acid, right? So that makes up nucleic acid. This is adenine. It's another nucleotide. Amino acid makes up what? Proteins. Told you I was off today. I stopped myself, though. I was like, that didn't make sense. Brain process, it didn't make sense, right? So that's an amino acid, right? It makes up proteins. Which does it have in it, sulfur or phosphorus?
Phosphorus.
Nope, methionine has sulfur. Proteins have sulfur because methionine and another amino acid, cytosine, contain sulfur. Adenine, which is our nucleotide, right, is the nitrogenous base adenine. The sugar, right, which is a ribose sugar, whether it's deoxy or regular ribose, depending on whether it's DNA or RNA. And then what connects the sugars to the phosphorus, right? Phosphate groups connect the sugars to the phosphates in each one. And remember, that's whose experiment did we look at that, where they actually radioactively labeled the protein, therefore the sulfur, and radioactive labeled the phosphorus, therefore the DNA? Hershey and Chase, with the bacterial phage, right? With the protein coat and the DNA on the inside. Okay, so you guys have kind of a general idea of size now, maybe a little bit better, right? So we definitely have a size issue as it comes to the compaction of our DNA. So they're saying, it's only about 2 to 4 nanometers in diameter. Right, we're not going to get insane about the measurements, right, as to one source to another, right? Just the degrees of change, right, that we're looking at. So we must tightly compact it in order to fit in the nucleus. And we do it quite elegantly, actually, if you ask me. So that whole DNA protein complex is referred to as chromatin. Right? So the actual name for our DNA, when it's complex with the proteins, which pretty much is always like that, is referred to as chromatin. Each individual one is a chromosome, right? So they do change, right? This is not a permanent association. It's an ever-changing, evolving through the cell cycle. But of course, the interactions with the proteins in the DNA determines the level of compaction, right, that we see for the chromosomes. So the first level that we look at is the nucleosomes. So this is where the double-stranded DNA, right, that double helix that we learned about last time, is wrapped around proteins. These proteins are referred to as histone proteins, and there are 8 of them that cluster together that the DNA wraps around. So it's referred to as its octamer, right? Oct meaning 8. Which is confusing because October is the 10th month in our calendar, huh? It used to be the 8th in a different calendar. That's where it got its name, actually. So the octamer is composed of 2 copies of 4 different histone proteins. So we make 4 different histone proteins. We stick 2 of each one, right? So 2 times 4 gives us the 8. We can take a strand of DNA containing about 146 base pairs. So A, G, T, C, A, G, T, C, right? Count out 146 of those. We will wrap that amount of DNA around this histone protein complex that we call the nucleosome. So there is a diagram of it there. Of course, this is very simplistic as to how it actually is, right? These molecules don't look quite like this. But they do have an overglobular structure, and a lot of them have these tails that interact with the DNA as well. So H is for histone, right? And then we have them numbered. And you'll see that we have an H2A and an H2B. We have an H4 and an H3. So where the heck is H1? It's not in the nucleosome. It's not part of this complex. We'll come to it. So you'll have segments in between, too, of DNA that don't interact with the nucleosome, right? That don't interact with the histone proteins. For me, it seems like this would take up more space than it does, huh? I know. It's like you're adding stuff. But think about this. When you're wrapping that cord for a device, or you're wrapping up that hose at home, you wrap it around something, don't you? Right? So at home, you may have that little stand, right, to wrap the hose around, right? A lot of times, you know, your cords and stuff for your devices, partly why even if you wrap it up, it tends to get tangled, is you don't have something to wrap it around, right? Does that make sense to you guys? So having these proteins, we wrap it around, it keeps it from getting tangled up, right? Keeps it organized. So in between this region will range between 20 and 100 base pairs. So when you put a whole bunch of them in a line, they almost look like beads on a string, right? There are a lot of times this analogy, right? We're taking beads and putting them on a string. This shortens the DNA about 7-fold. Okay. So if we've got 70 feet, say, we just went down to what? 10, right? 7-fold. Divide by 7. 10. So those histone proteins are mostly positively charged amino acids that make up those proteins. Why would that be? Why would we want positive? Why not negative? What do we know about DNA? It's negatively charged. It's negatively charged. Those phosphate groups, right? Those oxygens, they tend to be negatively charged. Overall, your DNA is negatively charged. That's why you can run it in a gel electrophoresis, right? And we want to attract it to the positive pole when we do stuff like that. So again, it makes sense that these would be positively charged, because if they were negatively charged, they would repel each other, right? It's like trying to put two magnets together the wrong way. They just keep flying apart. It's because you got the same poles pointing towards each other. So positively charged, mostly amino acids, so that it can interact directly with the protein. So this I don't expect you guys to memorize, but just so that you know, lysine and arginine are two amino acids that are positively charged. They're basic amino acids. So there's a lot of them in the histone proteins when you look at their amino acid makeup. And so the phosphates, as I said, are negatively charged. And so that's why they'll bind with each other. The histone proteins, for the most part, are a globular protein for most of it. But then they have these terminus, or these tails, right? So the amino acids coming off the end kind of do that. They just kind of trail off the end. They don't form secondary and tertiary structure with the rest of the amino acids. So there are 5 types of histone proteins. So as we noticed, right, we started out with 2A and B and 3 and 4. Where the heck did 1 go, right? So those are the ones that are in the octamer that make up the nucleosome. 1 interacts with the linker region, right? So that region in between the histones, and we'll see that in a moment. I mean nucleosomes. Shame on me. So this doesn't bind as tightly as those core histones that make up the nucleosome. And then, so they believe it may help with compacting the nucleosomes on top of each other. And I haven't decided, I don't know if we're going to go over the experiment. We've done enough experiments, right? Hershey and Chase. We might not go through the one in this section. So here, right, they just globbed all 8 of those proteins together, right? Histone 2A and B, both copies 3 and 4 in this globular diagram. Here, right, are non-histone proteins. You see these green ones? Right here, you see how the strand coming in and then the strand coming out? That's where the histone 1 proteins bind, is right at the nucleosome, but not within it. And it's binding to this, what we call linker DNA. So this is two different pictures, and I guess I should back up. Do you see how it makes two loops around? Did y'all see that? The DNA makes two loops around that core nucleosome. So it wraps around twice. And so in this side view here, do you see the DNA is color-coded in this orange and, I don't know, what is that, aqua, greenish blue color, more green than blue? You can see it on the outside here, right? But definitely in this view from the side, you can see that it's two strands. Do you see it? One strand here and one strand here. Here in the center view, you can see the proteins, and even they form helixes, right? Alpha helixes. I don't see any beta sheets in here, though. Right, this is more closely representative of what the molecules somewhat look like. 3D modeling, or 2D in this case, right? 3D would be cool. So we'll skip over this experiment, I think. But they use gel electrophoresis in it, right? So they're using electrical current to pull the pieces of DNA through this gel to be able to separate it out into the different sizes. It's kind of cool. So those nucleosomes, so beads on a string kind of thing, are then compacted into what's referred to as the 30 nanometer fiber. And they believe that the histone 1s really play a key role in this, because when they remove them, it doesn't do it. And they found at moderate salt concentrations, they'll remove the H1. And you see that classic bead on a string morphology. You see the histone, the nucleosomes along. At low salt concentrations, the histone 1 proteins stay bound. And so the beads are, they no longer look like beads on a string, right? They're associated together in a more complex morphology. So the picture says it all, right? So you see in this picture, see how they're little dots kind of on a string? So that's kind of like the beads on the string. This is without histone 1, right? So you just see the nucleosomes, right? These beads on a string. Here, histone 1 is still present. Do you see how it's much more compacted? So that compaction, going from nucleosome to 30 nanometer fibers, shortens it another 7-fold. Right? So 7 times 7, 49, right? Times compacted. About half the size it originally was. So actually discerning this structure has been difficult, right? Because when you take stuff out of cells, right, there's no way you can maintain how it was, or that exact environment when you extract it. So, there's still theories out there exactly what the 30 nanometer fiber looks like. So there's the solenoid model, and there's the 3-dimensional zigzag model. So this is the solenoid model, and this is the zigzag model where they're like kind of going back and forth. So for the solenoid, they believe it's almost like a spiral again. Does that make sense to you guys, where you take those beads on a string. So imagine taking the Mardi Gras necklace, right? And taking that necklace and turning it itself into another spiral. Does that make sense to you guys? That's what they believe the solenoid is like, if it's like that. Others believe it's kind of zigzagging back and forth and stacking on top of each other, like this. So there's two competing theories out there. Still don't have enough evidence to figure out. It might not even be either one of these, right, y'all? It might be something completely different. We need another Watson and Crick, right? And Rosalind's dead, so that's probably why we can't figure it out, right? So that's not it. Okay, but so far, right, 7 times 7 is about 49, round up to, say, 50. So we've compacted it half of its original size, as far as length goes, right? The third level of compaction involves the interaction between that fiber, however it's arranged, right, zigzag or solenoid or something else, interacts with proteins matrixings within the nucleus itself, almost like this scaffolding that happens. So that matrix is composed of two parts, what's referred to as the nuclear lamina. And so these are fibers that line the inner nuclear membrane. So right inside the membrane, there are fibers, proteins associated within the membrane, right on the inside. Then there are ones within the very core, right, the very inner center of the nucleus itself. So these are the internal matrix proteins. And they're not separate from each other, right? They're connected. But that fills in the inner space. So they believe they both have structural as well as functional roles. Right, but we're still, debating over that. So here's a diagram trying to represent some of this. So here, these proteins, you see these kind of light purple associated with the membrane. It's actually two membranes that make up the nuclear envelope. There's an inner and an outer membrane. And then there are, of course, pores in it so that messenger RNA can get in and out. Proteins can get in and out, and messenger RNA can get out. So that, as you'll see, where do they have it labeled? Is the lamina. Yep, nuclear lamina. So those proteins make up the lamina on the outside. And then the green proteins are the internal matrix, of all the stuff on the inside. So they believe they're fibrous, right? They're long proteins. So, oh, this is perfect. It's like when you open up the pumpkin, right? All those fibers on the inside. There you go. Thank you, Halloween. Everybody's seen that, right? That hold the seeds on the inside, all those fibers. Imagine something kind of stringy like that, where again, there's that concentration on the edges, even like inside the pumpkin. And all the seeds are suspended there. Beautiful matrix until it becomes a mess. So those 30 nanometer fibers, however they are arranged, they themselves even loop back on each other. And they interact with other proteins to interact with these protein filaments, these protein fibers within that matrix. So these are the radial loops, not to be confused with the domain loops that bacteria form, right? So in this loop, right, from here to say here, it can contain about 25,000 to 200,000 base pairs, just depending. And as they're showing you here, you know, there might be a particular gene within this region, but remember, that's nucleosomes interacting with each other to form the 30 nanometer fiber. And then you've got DNA wrapped around the histone proteins forming those nucleosomes. It's pretty compacted in there. So they believe that although there's certain even regions within the DNA that specifically interact with these proteins to bind with the proteins that make up the matrix. Not done. Right? So we got the nucleosomes, we got the 30 nanometer fiber, right? Then we've got the radial loops. So as those radial loops loop up along the fibers, each chromosome is located within a discrete, non-overlapping area, they believe. Again, this theory. But we've got some pretty cool stuff to show this. So here are each of the 7 types of chicken chromosomes are labeled in different colors, right? So how they did this, I don't know, but it's pretty cool, right? So number 1, you see it's in red. Number 2 in green. 3. I don't think they physically labeled it. I think they took a picture and then went in and colored it, by the way. So I don't think they could, but they can tell by the size, right? They know which ones are which. And like we can do with our chromosomes, one of the things we skipped over is like they can stain the chromosomes when they're in this compacted phase. And they get different bands on them. And based on that banding and the length of the chromosome, you can do what's called a karyotype. You can organize all the chromosomes based on which ones they are and do comparisons. Pretty cool stuff. So here, this is at metaphase, right? This is when they're the most compacted, right? And we're getting ready to line them up, right, and split them from one cell to the other in mitosis. Here is during interphase, right? So when they're not as compacted, right, when they're actually being used, when these chromosomes are actually being used. But you can see here, right, how they have it designated the different regions. So that, you know, they're you know, taking up a lot more space, the chromosomes, because certain regions are stretched out, right? They're not in the most compact form. Because the enzymes have to actually be able to get in there to read the information, right, to use it. So heterochromatin versus euchromatin. Well, hetero means what? Anyone know? Different. Actually means different. It's different. We're heterosexuals, right? Different sex. That's what that means. Opposite sex. So the level of compaction in interphase chromosomes is not completely uniform, right? You know, some of them got to be stretched out for that to be able to be read. So heterochromatin is the tightly compact regions, right? So when you look at a chromosome at interphase, those sections that are still compacted, we refer to as heterochromatin. It's said to be inactive, right? Because again, think about it. If it's really compacted, how are the enzymes getting in there to read that code? They're probably not. So can you guys think of a type of heterochromatin that women have in their cells all the time? Like really compacted, so much so it's given a special name. It's our X chromosome. Remember, we deactivate one of them completely. We don't use it at all. We have a special name for that heterochromatin, though. bar body. B-A-R. Bar body. It's the bar body. So radial loops domains are compacted even further. Right? So we've got the greatest amount of compaction for heterochromatin. Therefore, probably euchromatin is not compacted, right? Or true chromatin. It's less compacted regions of chromosome. Usually, of course, because it's less compacted, the enzymes can get in. It's transcriptionally active, right? RNA polymerase is going in there and is making messenger RNA in that region. The 30 nanometer fibers do form radial loop domains, right? But they're not as tightly compacted. So here's an example, right? Here's your 30 meter fibers, but you still see that they've got radial loops in here? They took out the proteins that they're scaffolding on, right? But you see the loops? But these are representing 30 nanometer fibers. So this is translationally active DNA, or chromatin, right? Because it's not just DNA, it's protein and DNA. Down here at the end, you see how it's much more compacted? The radial loops are even more compacted. In the middle and at the end. And of course, we have specific names on our chromosomes for these regions. You have the centromere, which are key to separating those chromosomes during mitosis and meiosis. And then you have the telomeres. Why do you think those are compacted? These ends. So what did we say? What did I just say about the compacted regions? What doesn't happen there? It's not transcriptionally active. Right? There's no genes there. There's no information being made into messenger RNA there. Same thing in the centromere region. This section of the chromosome, right, is transcriptionally active, right? It's much more stretched out. RNA polymerase can get in there. The telomeres are repetitive sequences. And if you know anything about how our chromosomes are copied, if we didn't have those end sequences, our chromosomes would keep getting shorter and shorter and shorter and shorter. And so we would lose information. So actually what happens is they get longer as we age. The telomeres actually get longer as you age. They add on to the end of the chromosome, as opposed to losing it. But because of that, it's not information we use. But it's important. So Afghan or knitted blankets, some of them, they'll have like those tassels hanging off the end. Okay? You don't want to cut those off, right? Because then the whole blanket would fray, right? They're tied at the end, right? And the reason why they could tie it was because they have that excess hanging off, right? A lot of people are just like, want to snip it. It's meant to be there for a reason, depending on how that was made. So the same thing for our DNA, right? If we didn't have those ends to basically secure the end of our chromosome, we would lose it. It would unravel. We would lose that information. And then the centromeres, as I said, are paramount for the proteins that bind to that region and pull the chromosomes apart during mitosis and meiosis. So again, that needs to be pretty consistent, that region, and we don't really want it messed with. So it makes sense that it would stay condensed, except for when it's needed during those processes. So this is our euchromatin, right, where the genes actually are transcriptionally active. It'll be compressed to the 30 nanometer fiber in radial loops, but it won't be even more compressed. Where the heterochromatin, where we're not transcriptionally active, which makes sense that our centromere and our telomeres would not be transcriptionally active. So conservative heterochromatin, meaning it's always condensed, right? It's always highly condensed regions. Permanently inactive, we don't use it. And again, as I said, usually contain a lot of those repetitive sequences, so we just keep adding on repeats. Go ahead, Tyler.
That doesn't say conservative. That says
Constituent, thank you, constituent. Which is almost like conservative, right? In other words, it stays that way. Constituent, conservative, it stays that way. Similar adjectives. I think I switch it out in my mind because it helps me remember that it stays. Does that make sense to you guys? So constituent, constitutive, like conservative. It stays. Facultative, on the other hand, I'll screw that one up. Facultative, you've probably heard that term before, right? If you've taken microbiology. It means what? Change. Change. So this is heterochromatin that doesn't always stay compacted. It can go from compacted to a little bit looser. So it goes between euchromatin and heterochromatin. But our telomeres, our centromeres, right, those are constitutive, right, chromatid. They stay compacted. So an example of one that doesn't stay compacted, per se, is the bar body, right? So remember, in our cells are going to pick, right, which one stays compressed. I forget when the picking happens. But when we are passing that on to our children, it doesn't stay compacted. But remember, that depends on species to species on how these things are imprinted as far as the X chromosome goes too, right? But for us, as far as the bar body goes, right, just because it's compacted in a somatic cell doesn't mean that it's always going to stay compacted, right? Our cells, as they divide, some point in embryonic development, it's chosen for those cells, right, which X is going to be compacted. But it's not a permanent thing. When we pass our X's on to our children, one or the other, depending for them. And for girls, in that case, is it going to be the one they got from mom or dad, right? Because that's how girls are girls. They get an X from mom and an X from dad. So I think, yeah, that's it. That's it for today. We're not going to go over section 10.6. So the important things, right, is what proteins actually make up that nucleosome. That's the first compaction. Nucleosome, then to 30 nanometer fiber. Exactly how that's arranged, we're not sure, right? Then the radial loops. And then the radial loops interact with the protein fibers within the nucleus, right, for even more compaction. When it's really compacted, we call it heterochromatin, right? When it's transcriptionally active and not as compacted as euchromatin, right? But heterochromatin can go between the two, right? But our telomeres and our centromeres stay compacted, though. They're constitutive, right? Compacted. Okay. I know, a lot of terms. Hopefully not too bad, right? So we'll get into the actual action with chapter 11. And I'll post that for you guys as well. I don't think I've added in the